Our first three lectures are going to be given by the laureates who are being recognized for the development of so-called next-generation DNA sequencing technology. So Shankar Balasubramanian is the Herschel Smith Professor of Medicinal Chemistry at the University of Cambridge and Senior Group Leader at Cancer Research UK's Cambridge Institute. He's co-inventor of the leading next-generation DNA sequencing technology, Solexa sequencing, now Illumina, that's made routine, accurate, low-cost sequencing of human genomes a reality and has revolutionized biology and, as we heard yesterday, is revolutionizing medicine. He's invented chemistry to decode several modified epigenetic DNA bases and DNA secondary structures in the genome, and he's made seminal contributions towards the understanding of their dynamics and function. His work on small molecule recognition of nucleic acids has revealed molecular mechanisms that can be exploited to modulate the biology of cancer. His collective contributions span fundamental chemistry and its application to biological and medical sciences. Sir Shankar was knighted in the Queen's New Year's Honour in 2017 and was awarded the Royal Society's Royal Medal in 2018. And in 2023, he was elected as an international member of the National Academy of Sciences, USA. So our 2024 Canada Gairdner International Award laureate, Shankar Balasubramanian, will be presenting Reading Your DNA. Please join me in welcoming him to the stage. Shankar.
Not a very good start. Thank you, Janet. Honored to be here and be a recipient of the Gairdner Prize, and thanks to the foundation. Can you hear me? Yeah, good. Okay, I'm going to give you my perspective on some of the challenges that we needed to overcome to develop the sequencing. I should say my part of the work was a collaboration with David Klenneman, who you'll hear from next, and it took place in the Department of Chemistry and led to the foundation of a company called Solexa, where the technology was developed into a fully integrated commercial system, and then Illumina acquired it and developed it further. These are actually other companies that have spawned out of very basic science projects in my lab. Now, Cambridge is actually famous for this structure, the DNA double helix, which in 1953, there were two important papers published. Watson and Crick did the interpretation, and Franklin and Wilkins generated the X-ray data. And the molecular basis for the genetic code boils down to this very simple relationship of the C base recognizing the G base through hydrogen bonding and the T base recognizing the A base. And it's remarkable, the whole of genetics boils down to this very simple molecular relationship. And DNA, of course, is a string of 4 letters arranged in a particular sequence. One copy of the human genome is made up of 3.2 billion of these letters organized in 23 chromosomes. And units of these letters that make up genes, these are typically thousands of bases. The genes encode for proteins that are the machines and hardware of cells. And that forms about 2% of the genome. The other 98% used to be dismissed as junk, but we now know that it's important in orchestrating and regulating how genes are activated. Now, the question here was the basis of the Human Genome Project. Why sequence DNA? Well, this blueprint provides the foundation for understanding living systems. Now, all living systems have a genome. That's the entirety of the DNA. Viruses, some viruses have an RNA genome. So sequencing it provides a foundation of knowledge. Now, one of the drivers for the Human Genome Project was to understand the genetic basis of who we are and why we're different, and why some of us are predisposed to diseases, and of course some diseases are caused by genetic changes to our genome. Now, many years ago, David and I paid a visit to the Wellcome Trust Sanger Institute, which is where the UK part of the Human Genome Project was taking place. Now, we had questions. At this stage, they were about a quarter of the way through their part of the project. But one of the questions was, to understand these things, you need to sequence at a population scale, not just one reference genome. Now, at this point, this was in the 1990s, it wasn't clear quite how that would happen. So we shared with them some of our thinking. Now, our start into this project has nothing to do with DNA sequencing. We were carrying out a rather abstract, pure, fundamental experiment. So you'll see David, and he'll explain the riddle of why he's holding a pint of ale in this photograph. And our postdocs, Colin Barnes and Mark Osborne, were key. Now, I'm a chemist, but along the way, I had gotten interested in understanding the chemical details of how enzymes catalyze reactions. And during my postdoc, I got interested in these wonderful machines called DNA polymerases that copy DNA every time a cell divides. And one of the beautiful properties of DNA is it can self-replicate, it can template its own synthesis. So we dreamt up an experiment. Now, David is a physical chemist, as you'll see in his talk, and single molecule fluorescence detection at room temperature was a relatively new observation technique at that point in time. So we wrote a grant to this research council saying we want to use this new observation technique to watch nature's machines make DNA and see what we can learn from that. That was our only aspiration. There was nothing useful implied in what we were proposing to do. We were just trying to see what we could learn. Now, the setup, they funded it remarkably, and I'm not sure they would fund that same grant today. Now, the setup included a glass surface, and we immobilized synthetic DNA. Here's the template, here's the primer. This oval is the machine, the polymerase, and we watched this strand being extended one building block at a time. And the way we did this was by using fluorescently labeled building blocks, and the fluorophore was the observation method. Now, after our talk with the people at the Sanger, and thinking more about what we were doing, we saw that what we were doing actually could be used to read the template strand of DNA by color coding the building blocks. So this is the concept behind what we did at Solexa. This same setup, here's the template, here's the primer. If we could persuade the polymerase to incorporate the correct base, now here T is the next templating base, here's where the building block goes. So you would bring this red dye with it. Now, if you could control this reaction so it would stop and persuade the polymerase to do it, you could then image this surface and you would read off a red dye, which would give you the identity of the first building block that's gone in. Now, if we could remove this dye and remove what was preventing the second building block from going in, we would be back here in the n+1 position, and in the next cycle, we could incorporate a T opposite an A, bring in this dye here, and read a second color. So cycle by cycle, you would see a series of changing colors at this single molecule. And in doing so, you were actually decoding the sequence of the building blocks in this template. Now, that's reading a piece of DNA that's attached to a surface. The way we were doing these experiments involved imaging lots of DNA molecules on a surface. Now, our vision for turning this into high throughput sequencing was extract DNA from a cell, fragment it into small pieces. Now, by controlling salt and dilution conditions, you can monodisperse these fragments on a surface so that each molecule of DNA would hit the surface and attach and stand in its own optically resolvable space. Now, you could then sequence all of these in parallel at the same time and retrieve a lot of information by scaling. So this was the principle that led to scaling DNA sequencing. Now, this was a crude calculation. It turned out to be dimensionally not quite correct, but the spirit of it was correct. So a human genome is of the order of billions of bases. That was the challenge. So if we assumed an array, and we assumed a rather modest array, we had a good understanding of the kinetics of some of these reaction steps. So we assumed a cycle time and calculated that you could sequence of the order of a genome on a time scale of days. Okay, so this was our prediction, billion base sequencer. Now, I'm going to tell you the essence of how this works and was reduced to practice in Solexa. So here's the primer, here's the template. The way a DNA polymerase works is it extends off this oxygen to incorporate the next building block. And I'm just going to show a little bit of chemistry here because I know there are some chemistry students at the front who might find this interesting. So this oxygen here acts as a nucleophile. This is a monomer. I've just put base. This depends on whether it's a G or C or T or A. And this oxygen actually attacks this phosphorus, and this diphosphate is a leaving group. And the enzyme, which I've simplified here, holds two magnesium ions in place, and they are actually the catalysts for this reaction. And you add one building block. So we had to hijack this process, and there were two aspects to this. One was re-engineering the building block into something unnatural, and the other, which I won't go into details about, was re-engineering the enzyme, the polymerase, to tolerate these unnatural chemical features in the building blocks. Now, there were two things that needed doing to the building block. One, this oxygen, which is the nucleophile for the reaction, we had to protect it on the monomer so that it would incorporate but not allow a subsequent incorporation, because we need to stop and acquire the information by reading the fluorescence. So we put what chemists call a protecting group on here. Now, the fluorophore we attach to the base at the edge that is not involved in Watson-Crick hydrogen bonding. So on C and T, we attach at carbon number 5. With G and A, we do what's called an N7 diazo replacement, and we attach there. Now, the chemistry here needed to be water compatible and mild, and a reaction that does not cross-react with DNA. And this was actually a lot of hard work went into this because it's a non-trivial problem. And it needs to be really efficient if you want to sequence lots of bases of DNA. Now, the chemistry of the first generation sequencing nucleotides developed at Solexa used this group here. This is an azide. Some of you will know of AZT, which was the first nucleoside drug against HIV-1. Now, AZT has an azide attached to the ribose, This is an azide and then a carbon and then an oxygen. And there's a reaction invented in the early 1900s by a chemist called Staudinger, where he showed that phosphines, which are these reagents, can reduce an azide to a primary amine. So an azide is a disguised primary amine. If you reduce this, this is called a hemiaminal, and in water it hydrolyzes spontaneously, like so. So we can cleave here, and we can cleave here, releasing the fluorophore with this reagent, the phosphine. Now, this is our intersection with what Pascal was doing at Mantea, and Pascal will say more of this. There was an important change to our original vision, which was single molecule sequencing. And in fact, we adopted this lovely method that Pascal and his colleagues developed. I won't go through the detail, but in essence, it takes a single molecule attached to a surface and then amplifies it into hundreds of copies of identical sequence. But they all stay in the same spatial location. This is hugely important. Now, the reason we looked to amplify fragments was single molecule detection is beautiful because it allows you to observe stochasticity. Now, stochasticity, unfortunately, was leading to errors in reading these sequences. And I'll mention his name because he's a former Gairdner laureate, the late, great Sidney Brenner, who often spent time in Cambridge. He once, he called me to his home in Ely, and I had a conversation with him in his kitchen of his house, in which he very forcibly said we should be amplifying these molecules. And in fact, he suggested a way, but really the best way of doing this is this method shown here. So this was adopted into the final sequencing system. Now, 2005 was around the time we did the first genome sequencing. It was a small genome, Phi-X174, which is a bacteriophage. And shown here is an expanded image taken by a charge-coupled detector imaging the surface at one cycle. And these round images here are actually amplified clusters of a single molecule of DNA. And the colors tell you what the base identity is for that step in the sequencing. So cycle by cycle, each of these spots would change color according to the next base that's been incorporated. And you can image transfer, convert, and digitize the information into sequence. And you get strings of sequences from each site on the surface. Now, back in the 90s, when we were trying to look forwards, one of the questions was, how long a piece of DNA could we read? Now, we did some calculations that suggested if we could read at least 25 bases, not knowing where the fragment came from, even if there were one or two single nucleotide polymorphisms, given the Human Genome Project was going to provide a reference sequence, we could computationally realign this to the reference to find out where it came from and cover most of the genome. So the challenge became to get at least 25 cycles. And you can see here the fragments get realigned and you can then read off the sequence. Now, all of that system today works sufficiently well to read hundreds of bases, and typically you read 250 from one end and 250 from the other end. This is called a paired read. Or today, I think it's possible to read over 500 bases in a single read. I think 600 is the latest system. Now, you can reassemble a genome. In 2008, there were 3 genomes published in one edition One which we were involved with was the first African genome, Yoruba genome. There was also the first Asian genome, and there was the first cancer genome. And they were all done at that time, reading 35 bases from one end and 35 from the other. It was possible to construct a genome. Now, this doubled the number of genomes deposited in the database in just one edition. 2006 was when Solexa released the first system. It was called a 1G genome analyzer. It looked like this, and you'll find these in museums now. They're already in museums, they've been relegated. But they sequenced 1 billion bases of DNA in a single run, which at that time took a few weeks, actually. It was not so fast. But it achieved the 1 billion base prediction or promise. Now, these higher-end instruments that were introduced by Illumina in 2021 had further optimized, engineered, improved the process at every level. And these beasts sequence trillions of bases of DNA per experiment. Overall, this is it's actually about a 10 million-fold improvement from the mid-90s when we were first trying to visualize this whole program of research. So over a million-fold improvement means sequencing the equivalent of about a genome a day, a genome an hour on these instruments, actually. You can sequence tens of genomes in 24 hours. And the vision of population scale sequencing is actually here. There are programs in many countries around the world where sequencing is being done at scale. This is one, the International Cancer Genome Consortium in the UK. Genomics England is a branch of our National Health Service, state-funded healthcare system. Other countries, Finland, the US, and of course Canada as well, have large-scale clinical sequencing programs. Now, I think David's going to say a bit more about application, but I'm just going to give you a taste in the cancer area. Now, this is a sort of a textbook example of a very specific mutation in a gene, BRAF. And this mutation causes the gene product to become overactive and drive cellular proliferation without control. And a certain proportion of people who have melanomas, skin cancers, have this very specific mutation. And there is a drug that acts on the protein and inhibits it. So patients that have this mutation respond very well to that specific drug. So this is an example of if you know the genetic characteristics of the tumor, you might be able to treat that tumor in a very specific and personalized way. These projects have generated and are generating enormous amounts of data linked to patient records. And what's emerging for some tumors is patterns of genetic changes that characterize the tumor and allow physicians to make an informed decision about treatment. But it's an ongoing project, and I think over the next 10 to 20 years we will see the full extent to which such information can help us deal with diseases such as the many types of cancers. I want to say something about early detection. Most diseases, and in particular cancers, it's desirable to catch it very early, and many of them are treatable with good outcomes if you can catch them early. And a burgeoning field is actually based on the remarkable observation that in blood there is cell-free DNA in the form of fragments circulating that can be retrieved from a blood draw, sequenced at great depth. And in patients that have tumor, the tumor cells necrose and release their contents into the plasma. And floating around are DNA fragments that originated from the tumor that can carry characteristic changes to the DNA, which may be either mutations or actually more commonly now, people look at what are called epigenetic characteristics of the DNA. And there are ongoing clinical trials now looking at patients and actually members of the population who don't show any signs of disease. And there are many cases of diagnosing disease. The holy grail here is to actually catch cancers at early stage, at stage 1. We still have some work to do, but this is an area to watch out for. This is my last slide. I'm just going to say something about what else I do in my, I was going to say in my work, but actually science is not work, it's a hobby and an obsession, and this is what else I've been doing. I've spent the last 30 years working on DNA, and I'm a chemist, so I'm always interested in the molecular aspects of DNA. And in that very first slide, I said to you, DNA is molecular information. It turns out that the order of GCAT in DNA is only one form of information that DNA stores and can convey. I'm going to give you 2 examples of areas that we work on. One is on structures. Now, I showed you the double helix structure of DNA. It turns out that is not the only structure that DNA can assume. And there is a quadruple helix structure shown here called a G quadruplex. And we've spent quite a bit of time elucidating these structures. They form in human cells. They tend to be very abundant in cancer states, actually, and they are indicative of cell states. And we recently provided evidence that they are part of the process that control the activation and inactivation of genes. So this is one area that we work on. The other is there's some interesting covalent chemistry that occurs naturally in DNA. You know, I alluded to a term epigenetics. Well, this is one of the 4 bases, the C base. And there is an enzyme that can put a methyl group here to form this methylated cytosine derivative, 5-MC. And this can actually be oxidized by another enzyme to this alcohol here. These modified bases exist in your DNA, in every cell in your body. And part of what these things do is, again, they are involved in orchestrating whether genes are switched on and switched off. And it's a grayscale, it's quite complex. And they help set the identity of cells. And this is part of why cells in your skin differ from cells in your kidney, cells in your heart, and so forth. They all share the same genome, but the epigenome reprograms them differently. And in the onset of disease states, you get changes in these epigenetic bases. So part of what I've been working on here is chemistry to sequence these modified bases. And I have to disclaim here a company called Biomodal that I'm involved with. They now have a sequencing technology that I helped develop that gives you a 6-letter human genome, where you get GCAT, MC, and HMC. And I predict, particularly in early onset of disease, that this is going to be quite important. So I'm going to stop there and give some thank yous. Firstly, the team in the university, superb team in Solexa, and also Illumina, who improved the technology further. Lots of people contributed to what I've just described, and I'm representing all of them, so credit to all the players. This all started with basic science. We weren't actually trying to do anything useful. And I think it's very important, especially for the young people here, to not be afraid of pursuing science for its own sake and ask curiosity-driven questions. You never know where it will lead you. I think it's also important for the politicians and funding agencies to acknowledge. I've already mentioned the team, and lastly, the technology has been used and applied by millions of people around the world, including those involved in clinical research, clinicians, patients have given their DNA for some of this work. So really, the impact and outcomes of this is a major team effort that hopefully will help solve problems. Thank you very much.